to lecture objective number 3, and we're going to compare the movement of molecules across the plasma membrane with carriers and through channels. So we're going to get into the other two types of passive transport. Now, again, I keep wanting to remind everyone because this is how questions will come on the exam. And this is one area that I know students often struggle with. So simple diffusion is not the same as passive transport. Passive transport is movement of particles down their concentration gradient. Simple diffusion is one type of passive transport where particles can move right through the membrane without the help of any carriers or channels. The other two types of passive transport do require transporters or carriers because some particles, like glucose, for example, is too large and bulky to move through the membrane by simple diffusion alone. So the second type of passive transport here is facilitated diffusion. This is where we're going to have the help of a carrier. And we describe the transmembrane proteins, if you can think back to that when we look at the structure of the cell membrane. And so these carriers are examples of those transmembrane proteins. And if you look at the image here, you can see that they span the entire length of that phospholipid bilayer. So they're going to be the perfect type of a protein to help particles to get right through that membrane onto the other side. So they're going to have a binding site for specific particles, and they're going to bind on one side at a time. So in the example here of this glucose transporter, this is called a GLUT4 transporter. It's bound into the membrane. It's permanently fixed in the membrane as we discussed, and then it's going to span both leaflets of the bilayer. It's going to bind glucose on one face of the membrane. It's going to undergo a conformational shift, and then it's going to kind of dump that glucose out on the intracellular surface of the membrane. And then it's going to go back to its original conformation. So this is how these carriers behave or work. They pick up the particle on one side, shape shift a little bit, and then enter the particle on the other side of the membrane. And while this is still passive transport, because they're moving according to a gradient, this is not going directly through the membrane. It's using a carrier instead. Now, let's look at the graph that shows us the movement according to facilitated diffusion. And what you'll notice here is that the factors that affect the rate of facilitated diffusion are going to be the rate of transport of each carrier. How fast can each of those carriers undergo that conformational change, dump the particle onto the other side, return to its original configuration, and then allow for more particles to go through. So the rate of each individual carrier will determine the rate of facilitated diffusion. The second factor, the number of carriers in the membrane. And here we can think about upregulating carriers or downregulating carriers depending on the state of the cell. So we can increase the number of these carriers in the membrane, and then we can decrease the numbers as well, and that can determine the rate of facilitated diffusion. Thirdly, the concentration gradient. Now, the concentration gradient is going to be to a point, right? And we'll see that the shape of our curve here is different than the graph that we saw for simple diffusion. Why? Because the carriers can be maxed out. So because we have a limiting factor in facilitated diffusion, when we get to a time where all the transporters, all the carriers are moving as fast as they can, they are maximally bound with glucose, for example, then we're no longer going to see a directly proportional relationship because our curve is going to flatten off because we are going to have to wait for new transporter carriers to become available in order to increase the rate any further. So the curve here is going to be linear to a point and then flatten off as we have maxed out or saturated all of our existing carriers. Okay. And that is to say that even if the concentration gradient continually gets exponentially larger, there's only so fast we can go with facilitated diffusion because of that limiting factor being the number of transports. Okay, let's talk about this example here. So glucose 4 transporter, the GLUT transporter 4. This is an example of a facilitated carrier. This is synthesized by the rough endoplasmic reticulum, or the rough ER inside the cell. They are packaged in vesicles, and then they are transported to the membrane to be inserted into the membrane. And the trigger for this insertion is insulin release. So when you have a glucose-rich meal, let's say you have a piece of cake or fruit, your insulin amount is going to be increased in the blood. As insulin levels rise, that is going to trigger the upregulation of GLUT4 transporters, and we're going to insert more GLUT4 transporters into the membrane in order for that glucose to be absorbed into the cells. Now, a very important clinical connection to this is diabetes. So diabetes mellitus is when there is a lack of insulin. This is especially true for diabetes type 1, where there's little or no insulin production. This can also be true for diabetes type 2, where there's insulin insensitivity. So your body has insulin, but it's not as receptive or as responsive to that insulin. So when we think about decreased amounts of insulin or insensitivity to insulin, that means that the number of GLUT4 transporters that should be inserted into the membrane in response to the level of insulin rising, that doesn't happen appropriately. So there's less glucose uptake into the cells, and there's more glucose being circulated in the blood. So this is where we begin to see the detrimental symptoms of diabetes as that blood circulates in, as that, glucose circulates in the blood inappropriately. It's going to travel to the kidneys and result in damage to the glomerulus, damage to the kidney membrane. It's going to travel to the eyes and result in damage long-term into the blood vessels around the eyes. And so this is why we begin to see the symptoms that manifest from diabetes, because we cannot actually utilize the glucose, so it sits in our blood and does lots of harmful, damaging things in the blood instead of being uptaken into the cells where it can be utilized for cellular respiration. So this is where the either lack of insulin, which is type 1 diabetes, or insensitivity to insulin, which is type 2, can result in the harmful consequences of diabetes. Okay. Now let's talk about the third type of passive transport here. This is diffusion through channels. And this is where we have particles moving through the membrane, but with the help of a channel instead of a carrier or a transporter. So these are also transmembrane proteins, just like the carriers are, but these are not as actively involved in the process. With the carriers, we saw that they bound that particle, underwent a transformational change, and then kind of pushed the particle out on the intracellular side. Here, the channels are not actively participating in the movement. So they are just standing as a passageway or a pore in the membrane and allowing for the particles to go right through. And these are also going to be substance specific. So only certain types of particles will be able to go through certain types of channels. All right. And here's what the channel looks like up close. So it's basically a polypeptide subunit that is bound to several other subunits that make up this more complex protein, which spans the membrane and acts like a kind of like a tunnel, if you will, or a passageway that allows the particles to go right through. The different types of channels are aquaporins. So aquaporins are channels that allow only water to go through the membrane. Again, water is a polar substance, so it's not going to be able to exist in the phospholipid bilayer. So it will require, for the most part, aquaporin channels to be able to go through the membrane. Ion channels are another type of channel that we see in the membrane, and this can be specific to different types of ions. So again, because we said that charged particles cannot go through the phospholipid bilayer, any type of charged particle, any ion, sodium, chloride, potassium, they all need an ion channel to be able to go through the membrane. And there are different types of ion channels. We have leak channels that allow ions to go right through without the help or any sort of gating or requirement needed. We have gated channels, which are sort of closed, and different changes can open up those gates. So the membrane potential change can open up those gates, etc. And then we have bidirectional ion channels that allow for ions to move in both directions. So there are different subtypes of ion channels that we're going to explore further when we talk about the neuron and the membrane potential, which is going to be coming up in Module 2. Now, the factors that affect the rate of transport of ions through different channels would be the rate of each channel individually, the number of channels in the membrane, and the state of the channel. So again, we can have a concentration gradient for sodium. Let's say we have a lot of sodium on the extracellular side of the membrane and a small amount of sodium on the intracellular side. So we have a concentration gradient. But let's say the ion channels for sodium are closed. Even though we have a concentration gradient, even though we have channels, if those channels are not open, then the rate of transport is going to be 0. So the state of the channel is extremely important, especially when it comes to the gated channels, because these are going to be open only under certain circumstances or requirements. And then if they're closed, there's going to be no movement of those ions. Okay, I'm going to skip over these as well and move on to our third lecture objective, which is to identify the 3 general factors that influence the rate at which substance can be passively moved or transported across a membrane. And then we're going to talk about the 2 factors that affect the rate of active transport. So we're going to really move on to look at the difference between active transport here instead. So what is active transport? We described it a bit earlier. It's non-spontaneous, it requires energy, and it's usually going to require or involve a pump of some sort. So this is usually some type of membrane protein as well, but this one is going to help directly generate the energy that's required to move particles against their concentration gradient. This movement can also be described as being uphill, or as we said, non-spontaneous. Okay. So in the scenario here on side A, we have the same number of particles dissolved as we do on side B. In the membrane here, we have a transporter, and this is called an active transporter. So what's going to happen is we can move particles from one side to the other against a concentration gradient. So here, the pump can move particles from side A to side B, and this is because we're using the energy that's harnessed in this pump to move particles, even though they're not moving along a concentration gradient. Okay, so that's the whole purpose of a pump or an active transporter. Now, what are the characteristics of a pump? One, we said it's a membrane protein, so it's going to be spanning the membrane, just like we mentioned for the other types of proteins. It's going to function as a transporter and an enzyme. So the transporter function of this is to move ions across the membrane. The enzyme portion of this pump is that it can catalyze the energy to make that movement by way of breaking down ATP. So it can break down the phosphate bond on ADP, liberate that phosphate, which has the energy in that third phosphate bond, and use that energy to move particles actively. So being a pump, it can harness that energy from ATP. And then just like with other types of protein carriers, it's going to have very specific binding sites. So it's going to be specific for some ions or particles that can be moved. It can also demonstrate saturation. So this is going to be the limiting factor that we mentioned earlier. As soon as all of those sites are bound, as soon as all of the pumps are maxed, then there's going to be a leveling off of the curve. In other words, there's going to be sort of a maxing out of the number of particles being moved or the amount of transport, because we no longer have any pumps that are free to move those particles actively. All right. Now, in this example, this is the sodium-potassium pump. We're going to talk much more about this very specific active transport pump. And this is found on most of our cells. So we're going to look at the way it behaves as an active or primary transporter. Now, when we think about active transport, there are two general types. There's primary active transport and then there's secondary active transport. So let's discuss the major differences. Primary active transport usually gets the energy from ATP hydrolysis. So going back to the sodium-potassium pump as an example, we bind 3 sodiums on one leaflet of the pump. Those 3 sodiums can move into the membrane by way of hydrolyzing ATP and releasing that energy. And then on the other face of the membrane, we can bind 2 potassium molecules. Again, phosphate can be hydrolyzed from ATP, and that will generate the energy that moves these 2 potassiums onto the opposite side of the cell, right? So the energy that is needed for primary active transport comes from the hydrolysis of ATP. Alternatively, with secondary active transport, the energy that's used comes from the movement of another particle that's moving passively. So going back to our initial description of active versus passive transport, we said that passive transport can liberate energy or donate energy. Active transport requires energy. So when we think about secondary active transport, we can move one particle passively, and we can couple that movement to another particle and use the energy that's harnessed from the passive movement in order to drive the active movement of another particle in question. Okay, so the energy that's released from ion movement can be harnessed to move another particle actively against its gradient. The example of this here is the sodium-linked glucose pump. This is an example of co-transport. So we're going to be moving sodium. Sodium is at a higher concentration outside the cell and a lower concentration inside the cell. So when sodium moves into the cell, it's doing so passively. Hence, the green arrow here means that we're giving up energy to do that, downhill or spontaneous. And we're coupling that energy by way of this sodium-linked pump and taking the energy that's harnessed to move glucose actively against its concentration gradient. Okay, so glucose here can be can be said to have a higher concentration inside the cell, a lower concentration outside the cell. So in order to move glucose into the cell, we're moving it against its concentration gradient, and so we need energy in order to do that. Another example of secondary active transport is the sodium-linked proton pump. And this is an example of counter transport. The reason for that is that we're now moving the particles in the opposite directions. So we're still using the same mechanism. We're harnessing the energy of sodium moving passively, but now we're using that energy to move protons in their opposite direction. So we're moving protons outside of the cell, using the energy harnessed from sodium moving inside of the cell. So this is still an example of secondary active transport, but this is specifically counter transport instead of co-transport. Okay, so here is a nice summary table, and I would encourage you to use this as a study tool. You can basically block out some of the descriptive characteristics down here and try to fill them in from your own understanding, right? Don't memorize them, but really try to understand the major differences in terms of the energy required, right? The fact that active transport requires energy, passive transport does not. You also want to think about whether or not transporters are needed. In terms of simple diffusion, that is not needed. In terms of all other types of movement, we do need some type of transporter or carrier. You can look at some of the examples of particles that can move by each of these means of transport. So fatty acids, oxygen, carbon dioxide can move right through the membrane. Charged particles like sodium, potassium chloride are going to need a channel of some sort. Glucose is going to need a carrier or a transporter. And then we talked about examples of active transport, right? Our sodium glucose linked transporter and our proton linked transporter as well, which would be examples of active transport. So definitely use this table as a way to summarize all the general types of transport under passive and active that we've talked about, and also know the different features. Again, you're going to see questions that come up on this and ask you to differentiate the different types as well as examples. Okay. Now, I want to end off here by just mentioning the way that this kind of all comes together. So while we think about active transport and passive transport, ion movement as separate individual types of transport, we want to think about how they are all happening simultaneously at any given time on any one given cell. So here we can see one cell surface has all these different types of transporters, right? We've got our sodium potassium pump, which is primary active transport. That is moving sodium out and potassium in. That is using ATP as the example. We have leak channels on the other surface of the membrane that is moving ions in the opposite direction. So while potassium leaks out of the cell, it can be actively pumped back in by the sodium potassium pump. Same thing for sodium. Sodium can leak into the cell according to its concentration gradient, and then it's going to be actively pumped right back out of the cell. So having both of these systems together, coordinating on any given cell, this is usually referred to as the pump leak system. So this is what keeps the relative amount of sodium and potassium the same. We never really see any net accumulation of sodium or potassium or any of our other given ions for that matter on any one side of the membrane. They're usually at the very same concentration in the extracellular fluid and intracellular fluid because of the fact that they are being moved in both directions according to their concentration gradient. Okay, so really important to think about how these are playing together and not just acting separately on the cell membrane.